Hello everyone, our chapter for today is protein synthesis in its two processes, transcription and translation. So let's go with the production of protein. Before we start with the real synthesis, we have to talk a little about the important tools that are needed for this synthesis. What are they? The important tools are RNA molecules. Let's look together what do we have here. So, the three tools that I'm talking about now are known as messenger RNA, ribosomal RNA, and transfer RNA. We have to put in mind that these three molecules are the molecules that are necessary for the protein synthesis. Let's describe the first tool, which is the messenger RNA. For sure, the messenger RNA, as you see, it's a one-stranded molecule made up of sequence of nucleotides. Okay, let me move to the second one, the ribosomal RNA. The ribosomal RNA is made up of two subunits, the small ribosomal subunit and the large ribosomal subunit. All the time I say that it looks like a burger sandwich where it has the big subunit and the small subunit. The third tool is known as transfer RNA. As you see, it has the T shape here. So, what can we say about this transfer RNA? It has an additional property. So first of all, as you see, it's folded in this way, and it has a 3 nucleotides at the binding site, and this is known as an anticodon site. In addition, it has an amino acid site where this transfer RNA is able to carry a type of amino acid. So now these are the three tools. We have to remember their names all the time: messenger RNA, ribosomal RNA, and transfer RNA. Now I have to talk about the origin of this messenger RNA. Where is it originated? Okay, so we knew that it is originated in the nucleus. Why in the nucleus? Because the RNA is known as a copy of a DNA molecule. So it carries the code of the DNA. That's why the origin should be inside the nucleus where the DNA is located. So the origination of this RNA molecule is known as transcription. So we are going to call this step transcription, and it is the first process of protein synthesis because the RNA molecule is going to be transcribed from the DNA molecule. How does this take place? Easily because of the help of certain enzymes known as RNA polymerase. The RNA polymerase is going to break down the hydrogen bond here, and the two DNA strands are apart, and from one of these two strands, the messenger RNA is going to be originated here or transcribed. So, and that's why it is a copy, it is a transcript, because it is transcribed from the DNA molecule in a process known as transcription. This is a DNA molecule with two strands, okay? Double helix, as you know. First of all, we have to name these two strands, okay? One of them is the coding and the second is the non-coding. Let's consider that this strand is the coding strand or the template strand. What do we mean by this? We mean that this strand is going to be transcribed into RNA under a process known as transcription. After being apart because of RNA polymerase, in a complementary way, the nucleotides are going to align themselves next to each other in a way they are complementary. So, if the nucleotide of the DNA is T, its complementary is A on RNA. And if it's A with DNA, for sure it's U, because you know that in RNA there is no thymine, and instead of thymine there is uracil. So RNA is a complementary molecule to DNA molecule, and as you see, if it's complementary to the coding, compare it to the non-coding, you will see it's the same as non-coding, but instead of T there is U. And here transcription is over and the messenger RNA is produced. Now the RNA molecule is produced. And as you see here, it's produced or transcribed in the nucleus. So in a step or a process known as transcription, the first process of protein synthesis. The RNA is ready and it has to be exported outside of the nucleus. Now we have to think about the second process in protein synthesis. So the code is ready. The code is found in the cytoplasm and it should be translated into a protein. Okay, still talking about two languages, nucleic language and the proteic language. So what are the other tools that should interfere here? Now it's the role of the ribosomal RNA and the transfer RNA. As you see here, it's a combination, it's an assembly between the messenger, the ribosomal, and the transfer RNA. All of them together in order to produce this protein. How does it start? As you see, the ribosomal RNA, they dissociate. The large subunit will be away from the small subunit, and the messenger RNA will split between them. So now the messenger RNA between the subunits. Okay, so which one will help now? It's the role of the transfer. Look for the transfer RNA, which is a very important molecule, which is a real translator carrying the nucleic language and amino acid language. So the language here, or the binding site here, is known as anticodon. It's made up of 3 nucleotides. For example, CGC. Okay? This anticodon is complementary to a codon on a messenger RNA. So this is a transfer RNA with 3 nucleotides, complementary to 3 nucleotides on the messenger RNA. Why is it important to be complementary? It's important because once this transfer RNA is complementary to this codon on the messenger RNA by its anticodon, this means that it carries the correct amino acid. Can't any transfer RNA bind any triplet or any codon on the messenger RNA. It should be complementary in a way it carries the specific amino acid. So the ribosomal, the messenger, the transfer RNA, the messenger between the 2 ribosomal subunits, the transfer binding the nucleotides, as we call them, codon of the messenger RNA, anticodon for the transfer RNA. And in this process, the amino acid will be collected. And let's continue. The ribosomal RNA as a very important molecule with details. This is the large subunit that we talked about, and this is the small subunit. So among them or between them, there is a site for the messenger RNA. As you saw, the messenger RNA, it is splits itself. It's found inside these two subunits. In addition, in the large subunit, there are two sites. The first is the P site, the second is the A site. Why are they important? If this is the site of the messenger RNA, so these two sites are for the transfer RNA. So I told you they made an assembly, and now you can imagine this assembly. So between the two ribosomal subunits is the messenger, and in the large subunits are the two molecules of the transfer RNA. Okay, so translation by itself, it's a very complicated process. It's not an easy one. So that's why we have to talk about its stages, about its steps that are three. So now we are focusing on translation with three steps. The first step is known as initiation, the second, elongation, and the third, termination. Initiation means that the beginning, the start of the production. So, the messenger RNA between the two ribosomal subunits, and on P site, the first transfer RNA will bind in complementary way, codon of the messenger, anticodon of the transfer, carrying the proper amino acid. Okay, but here there is a very important note. All the time, the first carried amino acid is methionine. So anyone, it starts with methionine and the codon is AUG. Okay, so then it has to grow now. For sure, you know that the protein is not made up of one amino acid. It's made up of more than one amino acid. That's why. Now the second one will take place and it sits in A site. Okay, so I have two transfer RNAs in P and A site. The two amino acids now are next to each other. Directly, a peptide bond will occur or take place between both of them, and the first bond occurs. Then what will happen? That of P will be away and A will take its place, and now A place is empty for a third transfer RNA to come. So the third transfer RNA comes and another peptide bond will take place. So 1, 2, 3, 4, 5, etc. It depends on the number of amino acids that belong to this protein. Okay, now the process has to be ended. It has to be covered. It's over. So the last stage is termination. What is termination? Termination is to stop translation. No more transfer RNAs, no more additional amino acid. Okay, this termination takes place when there is a codon here known as a stop codon, and it stops the translation, and now the peptide chain will be released and the protein is produced. Here I'm going to tell you or to teach you how to relate the amino acid to the code. You knew that the transfer RNA by its anticodon and the amino acid, it can carry this. Simply go for the messenger RNA. Each three nucleotides on the messenger RNA known as a codon. For example, the codon here is U on messenger RNA. So the transfer RNA, which can bind this codon, will carry an amino acid known as phenylalanine. Simply. How can we read it? Because it's a triplet, each codon is a triplet, it's made up of three letters. We read the first letter here, the second letter here, and the third letter, and we make a combination. I'm going to give you an example. If the codon is ACC, how do we find it? ACC. So this is A. Okay, it's here. This is C. And this is the third C. So here is ACC. So the codon ACC will have a transfer RNA carrying an amino acid known as threonine. And that's why we say that each transfer RNA has a specific amino acid depending on the codon. This application will help you a lot. So the DNA template, I divided them for you as triplets in order to be easier for you to find it. As a triplet, the first triplet in DNA is AAT. So the complementary on the messenger will be UUA. And the transfer will be complementary to the messenger AAU. Okay, I'm interested with the messenger RNA. Let me go to the genetic code table. This is the genetic code table. The first one is UUA. It's U, and here is A. So try to combine the three letters together. A. So the amino acid is leucine. Here is leucine. The second one on amino acids on the messenger RNA is GCA. G, here C, here A, GCA, so it is alanine. And you continue, arginine, threonine, and histidine. Okay, so by the messenger RNA, each triplet is known as a codon. The three letters of the codon U, combine them with the genetic code table in order to find it. Here another application in order to know how to terminate or to reach the stop codon. Okay, as you see here, the messenger RNA, it's made up of certain codons. The first one, leucine, the second for alanine, the third arginine. Let's go to UAA. This is U, this is UAA, this is U, and this is A, and this is A. So the codon UAA codes for stop. So the meaning of stop means that there is a termination and the protein synthesis has to stop and the peptide it's ended here. So it's made up of the previous amino acids and no more continuation. And in this way, we covered the chapter.